What I wanted to do today is to really talk about HiFi reads and why we think these really are going to be or are the future of sequencing. Really, we haven't had a data type like HiFi before, something that's long and accurate. And so I'm going to spend the first sort of 20, 25 minutes going through the HiFi data type, some vignettes of how it's being applied. And then I'm going to throw it over to the wonderful Melissa, who's going to talk all about her experience with HiFi and getting set up in a lab. So for those that have, obviously this is a virtual environment, things going on at home, be ready for Melissa's talk in about 20 minutes. That's the important part of today by all counts. So firstly, since we can't be there, in person, unfortunately, we are doing this virtually. So I wanted to start with a poll, actually, to prove that this is live, it's interactive, and try and engage the community. So you'll see on your screen a poll should have popped up. Really want to know from you guys, what is it about running a core lab that keeps you up, keeps you thinking and worried at the moment about the state of the affairs? I see some results coming in. Given the lateness of the day, we'll give it just a maybe 10 or 15 seconds for it to come through, and then we'll look to close it out. I can see a few more responses coming in at the moment. Okay, why don't we pause it there and look? I think this aligns pretty much exactly with what we saw in this morning's session. This idea around meeting or exceeding customer expectations being just such a critical part of the role. And look, certainly having run small service genomics offerings and core labs, I completely understand. So that's not too surprising. Running the multiple technologies one is certainly something that came up this morning, but seems to have come up a little stronger this afternoon, which is good. And I think you'll start to see some examples, especially around the meeting the expectations of customers and what you can do and how the different data types can come together to really help out. So I'm glad the poll system's working. So let's keep going here and look along those lines of what can we do as PacBio to help you overcome some of these concerns, these challenges that you guys face on a day-to-day basis. I really wanted to call out where I think, having come to PacBio, I'm clearly a huge believer in the technology and the data type that we have in HiFi, but just call out some of the things that I think really can help you and your lab be successful and drive your business, around the huge demand we're seeing for HiFi sequencing. So I'll spend a bit of time talking about that. Obviously, the commercial side of the company is growing, and you've heard that from many of our leaders in the company, and how we continue to offer outstanding support. And really, you'll see with some of the product announcements that we've had recently, trying to make the system and the data type as accessible as possible and lower some of those challenging barriers to entry around things like compute costs. At the end of the day, it really is about how do we make the data type that PacBio provides as accurate, as affordable, and ultimately as essential as possible for your work, and ultimately for your customers' work, so they can get those results that you guys help them with so often. If you think about that idea of how do we answer questions and what is the right technology, as I said earlier, HiFi reads is a data type that we really haven't had before, and I think of it in sort of 5 key kinds of areas. It does give us those long reads in the tens of kilobases, and we can sequence molecules from sort of as low as 500, or I think Melissa will even mention some runs with even shorter molecules, all the way up to things that are greater than 50 kb. But at the end of the day, those molecules are great because they are so highly accurate. They're free effectively of systematic errors. And because of the HiFi data type, we can remove what little error is there to get to a really high consensus accuracy for you to take forward into your analysis. We still maintain that single molecule resolution, which is really amazing. And every single HiFi read is greater than 99.9% accurate, so better than Q20. And it allows us to have that uniform coverage because we aren't doing DNA amplification. We get to reduce some of the bias that we see in the context of the sequencing. And then finally, something that we have continued to do and provide, and is I think a unique feature here realistically, is the ability to continue to detect epigenetics within the data type as well. So we can really start to look at that. And there's been some really great papers recently. I think Dennis Lowe just put one out showing how they could use HiFi reads to really extract out epigenetic information as well as everything else that comes along with the value of longer reads. So on the right, what I wanted to show is sort of how do we get to HiFi reads? I'm sure, you guys as technologists and folks that really understand the needs and the products better than really anyone, really understand and appreciate this. But I just wanted to step through it quickly because it is really a nice way to describe what makes HiFi reads so special. And it all starts with that long, high quality double-stranded DNA. We then ligate on our special SmartBal adapters and size select for molecules in that sort of 10 to 20 kb size range. Once we go through our annealing of primers and DNA polymerase binding, we're actually able to sequence in a circularized fashion. So we can go round and round that molecule multiple times. And this is where the HiFi part kicks in. By going around multiple times, we're actually able to create a consensus that removes all of those errors effectively and leads us to these ultra high quality reads. And I think what's really exciting is if you actually start to look at those reads, and this is data that's come from mostly the Precision FDA challenge that we'll talk a little bit about in the future, but also some recent updates from Oxford Nanopore. Those reads of, like I said, every read is better than 99.9% accurate, but you do see here that we're really hitting up to 99.95% accuracy on the HiFi reads in green, surpassing even Illumina NovaSeq short reads, and really differentiated away from the ONT reads here using the Bonito pipeline, which are coming in at sort of 98.3. But with that really big distribution as well, which is important to notice. The HiFi reads, as I said, all cluster right around that ultra high data, that ultra high quality. So for the first time, we have not only long reads, but also really accurate reads that we can take forward to handle these into this analysis. And that really does play out when you think about all the things I just mentioned around the accuracy, around the coverage, the completeness, our ability to look at long range phasing and really get, honestly, the best performance for all variant classes. Going back to the Precision FDA challenge, you can see on the bottom right here, when we look at those data sets, the total number of errors that are showing up in those data sets for SNVs, indels, and SVs on PacBio is incredibly low. It's almost 6 times lower than the Illumina data and about 30 times lower than the ONT data, which is truly amazing. If you look at that truth challenge, it's pretty impressive. PacBio's won across the board for a single technology. So to get any better than that, you really do start having to bring together multiple technologies, and you do end up in an area of somewhat diminishing returns. So to prove that, again, this is live and keep you guys engaged, we've got one more little quiz here just to make sure you guys are following along with me. So what are HiFi reads? And again, we'll just leave this open for sort of 15 or 20 seconds. Hopefully, folks will be able to respond pretty quickly on this one, if I've done my job right, that is. Fantastic. I mean, I think we can probably close that one out. Everyone seems to have selected the highly accurate long reads that are actually quite affordable. So seems I'm doing an okay job of describing these, which is good. I did have someone this morning mention that it was books for audiophiles, which led to an interesting conversation. But no, I'm glad you guys have kept up with the HiFi sequencing. Okay, so we've spoken about HiFi and the value of it and what we can do. I wanted to go back just towards the end of last year and really call out the Sequel IIe system, because I think this is something that has really helped with the accessibility of this. And Melissa will do a great job talking about the benefits to her lab. But with this system, we are able to get HiFi reads directly out of the sequencer. So no post-sequencing processing on a cluster, for example. All happens on the instrument, which is truly pretty amazing. And because we're collapsing down all those sub-reads to get to that one HiFi read, we can actually reduce pretty impressively, about 90% reduction in the data storage needs for a run. And that in turn leads to a reduction in data analysis time. because we have obviously less reads going in, because we've collapsed them down, and they're at a higher quality. It makes for the analysis much easier. It's also cloud enabled, but if you were to sort of have to run these through at a pretty high rate through the cloud to do all this processing, that could be costing you sort of around $700 a genome. So this platform has really changed the landscape and is really quite enabling and accessible when it comes to getting that fantastic HiFi data out of the system, and as simple as possible for you guys to use and return to your end users. So what I wanted to do now is sort of shift gears a little bit and just talk about a couple of different areas of sequencing where we're seeing HiFi reads applied. And obviously it's still pretty early days for the data type, but we are seeing really fantastic uptake. And so what I wanted to step through was just, as I said, a few little different vignettes here in where HiFi can be used. And this is actually taken this table here from a really nice single pager that the team has put together that talks about what can you do with a single smart cell, and a single smart cell being about $1,300 US list price. It's consumables only, as you can imagine, but it really does start to open up the market in terms of what you can get out of that single run. And so I'll start talking about de novo assembly. So obviously we can apply HiFi reads to assemble various organisms. You can see on the left a pretty large assembly that was done on PacBio HiFi. So the redwood genome, large in every sense of the word. It's 33-fold coverage and assembled to about 48 gigabases in size. So a really impressive assembly there from the HiFi data type, actually far exceeding anything that was able to be done before. And then on the right, you can start to see some population-specific references that are coming together. And building on this theme of how easy the data is to work with and how efficient, having those ultra-low error rates and the long reads lets us get to sort of 5.5 hours to assemble sort of a 20x human genome. So then you end up with high contiguity, fully phased haplotigs, high per base quality of these final assemblies. And as I said, because you've got that long, high quality data, it is really quite easy to use. So we're seeing an increasing number of these programs come through. There's some really great talks last week at AGBT around exactly this, from the telomere to telomere and the Human Pangenome Reference Consortium. But obviously it's not just in the human workflow. We are seeing this applied to things like the Darwin Tree of Life program being run out of the Sanger along with collaborators, as well as some of the other programs mentioned on the right there, like the Earth Biogenome program. I think what's really important to call out here is that while we have a sort of standard approach that needs sort of 3 micrograms of DNA per genome, we do have alternative protocols that really have helped these specific programs get up and running, knowing that you're not going to be able to always get 3 micrograms of high quality DNA out of some of these organisms. So we have a low DNA and even an ultra low DNA that lets you get down to just 5 nanograms of input there. It is amplification based, but for those cases where DNA is very hard to come by, it's really fantastic to see that we can still apply these library prep processes to the HiFi data type and achieve pretty fantastic results. So shifting gears a little bit on from there into sort of more of the variant detection world, we sort of already spoke about the precision FDA results and how impressive the data type was in terms of getting very low error rates, but I wanted to sort of put it into a different context, more around the rare and inherited disease. And I'm sure it comes as no surprise to folks in this audience. There are a huge number of rare diseases that when you look at them in aggregate, they really are affecting a very large percentage of folks. Sort of 1 in 10 individuals are impacted by a rare disease. And we believe that about 80% are genetic in origin, yet greater than 50% of these remain unsolved through the current processes of short read exome or even short read whole genome. And then even in the Mendelian space, we know there's sort of over 8,500 disorders, and yet only 40% have about 40% have an unknown genetic cause. So our goal here really is to show that through the use of PacBio and HiFi, we really can start to shift these numbers and make a real positive impact. And this sort of tracks with what's been happening over the years throughout the evolution of the technology, where we started with things like karyotyping and had a pretty low explanation rate here as to the causal variants that were happening in these cases and diseases, moving through microarrays. And as I said, even short read sequencing at the genome level is still coming in somewhere around 40% explanation rate. So it may be that you can process a large number of samples, but at the end of the day, if you can only solve 40% of them, that's going to remain pretty challenging. We're, again, pretty early in the adoption of HiFi reads for this space, but being able to call at very high accuracy SNVs, indels, SVs, CNVs, and all the other kinds of things that happen in the genome is really starting to give us some insights into how much more we're going to be able to explain using this data type relative to others. So we've seen pretty great adoption and a number of publications and press releases come out. Folks from Children's Mercy teaming up with us to talk about their fight of rare disease using HiFi data. Even the All of Us program adding this to their program, and Hudson Alpha using this as well. Neil Miller from Children's Mercy, again at AGBT last week, gave a pretty great talk, just showing how far the HiFi data type was allowing them to go and solve cases that were being left unsolved with other technologies. What we're seeing, and again, early days, is somewhere around sort of 2/3 of cases are now getting solved. So really pushing us over that sort of 50% mark using HiFi instead of alternative technologies, which is pretty amazing. So between this and the fantastic results we showed from the Precision FDA, it really is highlighting that the long, highly accurate reads are incredibly valuable for these sorts of studies. Okay, so moving on to transcriptome, certainly a huge area of work for many folks in the audience, I imagine. There is an inherent benefit of being able to sequence the full length of the transcript and really see all the splice junctions, alternative splicing, et cetera, that happens, and not have to make those assumptions. So, inherently, the HiFi reads allow us to get all the way through an individual transcript and really see these without making those sort of assumptions, as I said. And actually, we've seen some really great papers coming along that describe even the use of full-length isoseq at the single cell level. So here in this publication, Gupta and colleagues were really able to identify certain expression patterns and allele-specific expression realistically through different types of tissues here, from neurons through microglia, etc., and showing just how important it is to be able to sequence through and see all that alternative or allele-specific expression happening. Okay, so pivoting away from RNA to amplicon sequencing, this is one that clearly, given the context and where the world is today, amplicon sequencing of COVID has just become a huge opportunity and a huge gain for the world in really tracking and understanding through surveillance the use of genomic sequencing in the context of COVID. And so there's a number of publications, but recently we have put out a collaboration protocol here with the folks at LabCorp, really talking about how you can apply this to PacBio sequencing, allowing you to sequence almost or up to about 1,000 genomes per smart cell with really high resolution and get to complete genomes. And this has really resulted in some pretty amazing things being discovered. And we're seeing a real uptake of this method. Just a few weeks ago, when we looked at the submissions to GISAID, we were able to see that there was almost 50% of what was going in there was PacBio data. So similar to the number of short reads. And obviously with the long reads, there's a number of things that we can start to detect. Obviously this South African variant, I think, was first discovered through LabCorp's process, which is the protocol I just mentioned. But it does allow us, this protocol, to really address some of the challenges that are happening. So because we're using larger molecules or larger amplicons, we can get better balancing of the primers for the multiplex reaction, resulting in sort of fewer dropouts. It allows us to really look at all the variants. So we're seeing some pretty large insertions and deletions happening. As I said, you can scale up to almost 1,000 individual samples on a smart cell. So for that $1,300 of consumables in sequencing, you can get through a huge number of samples there. And it also allows us to be pretty flexible in the batch sizing. So a really fantastic sort of workflow that really highlights the benefit of the HiFi data type in the PacBio system. I mean, you can imagine that this is going to be a huge area going forward. I think the world is really aware of the value of genomics when it comes to these sorts of events. And so things beyond even just pathogen discovery and epidemiology, and even heading into sort of immune escape, are going to be key factors going forward. But again, that very high data quality, that unbiased nature we get from the sequencing, and the ability to really look across long reads is truly a key part of this and going to be very important, I believe. So just to start wrapping us up, because I know we're coming up on time, and I want to make sure Melissa has plenty of time and we can do question and answer afterwards. I just wanted to touch finally on microbial sequencing. Again, something I think that many of you in the audience are clearly involved in. And so there's a couple of things here that have recently come out. Firstly is a new 16S protocol from Shoreline Bio. This really allows us to sequence sort of 192 samples, full length 16S on an individual SMRT cell on the Sequel IIe. Pretty inexpensive to get up and running, and really allows, when you think about the complexity we're talking about and the sequencing cost, to get down to something around $8.50 per sample, which is pretty amazing. So certainly getting down towards the old Sanger sequencing prices there. Beyond this, there's also a new strain ID kit that uses a larger amplicon. It's about 2.5 kb that actually spans the 16S, ITS 23S region. It allows for strain level identification and comes bundled with a really nice software suite that allows you to sort of create these identifications and do strain tracking across various experiments. So some real nice advances there in the microbial space. And again, just to sort of highlight that, this HiFi data, especially for metagenomics, is increasingly important. The long reads really do allow us to capture a pretty significant number of genes in each of those reads. So being able to keep those things phased and together is really impressive. And finally, I would just say that there's been some real nice advances in the assembly sides of things as well. So you can see on the right here, some assemblies from a gut microbiome sample. And the HiFi data type is really allowing us to get to some pretty amazing and complete assemblies. You can see the circularized assemblies there on the right as well. So with pretty low coverage, we can get to some really impressive results, which I think goes back to this idea of how can we make sure that your customers are really getting the best result for their experiments that are obviously working with you. So as I said, HiFi as a data type is relatively new, but we're seeing it applied across a huge number of different applications, and we're seeing the benefits of those really high quality long reads coming through in the simplicity of the analysis and what you can really get out of the experiments. I would be remiss if I didn't mention one of the promotions that the team is running at the moment to sort of trade in one of your older either short read or even other long read technologies to really get you access to a Sequel IIe and get access to those HiFi reads that really do provide some of the best outcomes we're seeing in these experiments. So with that, I'm going to wrap up with just one last question that I think is really important, and that really is, who's excited for Melissa's talk coming up next? So I'll leave this one open real briefly because I'm pretty sure I already know the answer. All right, it seems unanimous that everyone is excited for your talk, Melissa. So why don't I go ahead and pass the ball, and we'll come back for questions. And just as a reminder, if you post those in the chat, that would be great. So thanks again.
Great, thanks, Dave. I'm going to stop your screen sharing and start my own. And thanks to everyone at PacBio for their invitation to speak today. I'm excited to bring this presentation to this session. So as Dave mentioned, I'm going to talk today about my journey in bringing PacBio to the Bluegrass State. I'm migrated here from New York last July. So I've been here about 9 months now, and here being the University of Louisville in Kentucky. So when I was in New York, I was the Associate Director for Technology Development at the Icahn School of Medicine at Mount Sinai. There, our philosophy was to use novel sequencing data types and advanced instrumentation to explore complex problems that were underscored by complex genetic loci. We did that with a lot of different instruments. In fact, I'm going to put the list up here on the right, and it's not to make everyone drool, but it's just to drive home that this was an incredibly amazing environment to work in. I learned a ton about all the different technologies. It was also a very high pressure job. We processed over 10,000 samples a year across these different platforms, and often a 7 days a week, 24 hours a day type of job. We were very lucky to have incredible support from the administration in order to keep this lab up and running and get access to all these technologies as they became available. But I think similar to almost everyone I've talked to, 2020 was a year of change for everyone. Our team worked very hard to push out this publication in Science in May of last year, documenting the first incidence of SARS-CoV-2 in the New York City area and tracing that to a European origin. And this is actually the first paper to use long-read sequencing to sequence SARS-CoV-2. I think in the course of all of these events, I think, everyone that I know at least has done a lot of rethinking about their priorities in life, and that wasn't any different for me and my family. And given, our desire to be closer to family, which are in the Midwest, and to strive for a little bit more work-life balance and maybe at least a shorter commute than we had in New York, as well as the incredible fortune of obtaining some successful independent funding for research projects that I launched from the lab in Sinai, I had the opportunity to move on from New York and come to Kentucky. So not the most typical of migration patterns, especially sometimes in science, but this was an amazing opportunity at the University of Louisville to start my own lab, as well as engage in a lot of collaborative research. So as I was starting to go through that process, I had to think about what the Smith lab looks like in my mind and develop a philosophy for sort of my ongoing research. And my philosophy throughout the 20 years I've been doing research is that health and disease don't exist in a vacuum. They are the result of complex interactions between altered self or pathogen infection, the genetic context and immunology within the host, as well as external and microenvironment influences. And I've worked in all of these areas to some extent or the other over the course of my career. And when I learned while I was trying to form what that would look like in terms of establishing a lab, that all of these questions, at least in the past 8 years of my research career, have been informed by smart sequencing. And so part of my process of moving here was to ensure that we would be able to have a PacBio on site. Now, this is a pretty large investment for a university, not only monetarily, but also philosophically and sort of a vote of confidence for a university that has not had this technology on site before. We do have a small genomics core that runs a few of the lower scale Illumina instruments, as well as some 10x instrumentation, but single molecule sequencing is a pretty far stretch from those instruments, at least philosophically. So I was very lucky to have 3 big benefactors here at U of L, my department chair, Dr. Ron Gregg, our executive vice president of research and innovation, Dr. Kevin Gardner, as well as my very close collaborator, Corey Watson, and I'll talk about the work I do with Corey a little bit later in the talk. But together with their support, we were able to bring on a Sequel IIe to campus as part of my recruitment last year. And when I was doing this, it became very apparent that there's a lot of additional things that have to be thought of and challenges that have to be addressed when you bring smart sequencing to a new location. So the first, of course, is the instrument itself, space, staff to run it, any ancillary equipment. So we were very lucky, again, as I mentioned, there is a genomics core on campus, as well as a core that's very focused on Sanger sequencing and genotyping. And so I merged with that DNA core and luckily was able to share some of the trained staff there, which has been an amazing boon for my group. Additionally, ancillary equipment, this is something that's not discussed quite a lot when you're talking about the big purchase of the sequencer itself. But you do need a compute infrastructure capable of handling the data. That will be its own separate bullet point. It is its own beast. But you also need a lot of the QC equipment necessary to ensure that you're generating the best libraries possible for sequencing. And for us, that was the fragment analyzer from Agilent now, as well as the size selection systems from Sage Science, like the Blue Pippin, which thankfully for us already existed on campus, but are also very necessary for generating those really nice HiFi libraries to get the kind of data that David was just reviewing. Compute, as I mentioned, is its own beast. And I think, you think about the data quality that David just went over and the amount of data you're generating per SMRT Cell, that eats up or requires, I guess, a lot of compute performance in generating those data, as well as the storage required to save them, both for personal research projects as well as service projects, where, as you all know, running core providers, no one ever downloads the data the first time around, and often they come back 3 years from now when they publish and want to download it again. So we need to make sure that that data will be accessible for long periods of time in a way that is cost effective, at least as much as possible. In addition, the software for the instrument runs on a Linux operating system, not something that I was typically trained in as a molecular biologist. So finding personnel who can install, manage, administrate the compute environment is also critically important. Now, we have a great Cardinal Research Cluster on campus. However, it is packed with other people using it and somewhat limited due to its age and its ability to mount some of these newer tools. And so we had to work around the existing cluster. In fact, I was able to obtain a local dedicated server on site. It's actually right directly behind me under my desk. We've been working with Microsoft and Azure to create a dedicated blob storage for data transfer to our customers that is transferred securely. But really what I'd like to hammer home and underscore David's point is that we upgraded to Sequel II earlier this year, and it really has changed everything for us as far as tractability and affordability of maintaining the data here, especially because we're doing a lot of workarounds to our existing cluster. So these data files that are generating 10x HiFi coverage per genome, before HiFi was generated on an instrument, you would download the subreads, process those HiFi reads on your server before exporting those to your customer. Those raw data files would often exceed a terabyte of storage space. And once the CCS is performed, you collapse that terabyte into 50 to 100 gigabytes. And so now all of that processing and that collapsing happens on the instrument. I don't have to pay for that compute, aside, of course, from the upgrade. I don't have to store that raw data because it's the HiFi reads that are exported off. I don't have to queue up in a shared cluster environment to wait for my chance to access that processing power. So it really has changed the scalability and availability of us to really maximize our instrument while still maintaining support systems for managing the data. The last two things I want to point out are things that sort of happen as you go, but that are definitely important to think about. So project management, balancing internal and external projects, as well as billing. We do this using cloud-based platforms like Smartsheet and QuickBooks. This is really important. We have a small but mighty team now, but we really don't want to be bottlenecking on just me to press every button. So having these cloud-based platforms allows us to chat back and forth, create invoices, send out bills in a shared way that allows things to get done and not back up on one person. And lastly, of course, marketing and awareness. We are the first PacBio system in the state of Kentucky and the first in several surrounding states. And so it really not only is the work that we know we can do, but getting out the word of what we can do to enrich the research around us. So I've taken the opportunity as today to participate in every webinar, seminar, and conversational opportunity possible to educate my peers in the local region of what is now possible with this system on campus. And once I started doing that, there's a tremendous amount of opportunity that opened up that maybe wouldn't have been so apparent out of the gate. So the first, of course, are existing collaborative grants and projects that we have, myself and Corey, together. Also, U of L has been very fortunate to receive several COBRA awards. So these are awarded for large center formation in the IDEA states. They're quite long and quite well funded. We have a Center for Cancer Immunology and a Center of Functional Metagenomics. I foresee a lot of very productive collaborations with those investigators and our core. Additionally, we're part of the INBRE, or IDEA Networks of Biomedical Research Excellence Network. The goal of INBRE is to offer access to cutting edge technologies, both informatics and molecular processing, to smaller researchers at smaller schools that may not have their own genomics core or something like this, and at a reduced or internal type pricing scheme. And so we are working very hard to integrate our core into the Kentucky INBRE network and are actively collaborating with INBRE investigators. I think this is one of my favorite parts about being in Louisville, is really being able to sort of spread the PacBio love around. Lastly, of course, it shouldn't surprise anyone, but maybe me, that now that I'm in Kentucky, I'm sequencing a lot of horses. So we have a collaborator at UK. We're doing quite a few equine projects with him. We've developed a lot of collaborations at West Virginia, at UVA, as well as UT Health. So we're really excited about this new network that we're building. I think there's a lot of cross-pollination that can be done across a lot of fields utilizing that PacBio instrument. So not only are we now the Smith Lab, but we have become and evolved into the University of Louisville single molecule sequencing core, whose mission is not only looking at host pathogen interactions, but supporting genomics research at U of L and beyond. So my lab, we have a small group I'll show you in a second, of initiated independent research projects, including novel assay development for both virus and immune system components, as well as the generation of several high quality reference genomes and transcriptomes as part of collaborative projects. But what I'm here to talk about today, of course, is our work as a Sequel IIe service provider. I think we are the most recent service provider added to the family at PacBio, and we're delighted to be there. Before I go any further, I'd like to make sure I acknowledge my small but mighty team. I'm very lucky to have found this group of people. They keep me sane on a regular basis. We're still all working at the bench side by side while things are getting up and running. Elizabeth Hudson from the DNA Core has taken on a large amount of the HiFi projects. Her hands are amazing. Andy Lauer is my lab manager and has definitely kept me above board on all compliance issues, which can be challenging. And Allison Silver works between Corey and my lab, managing a lot of the immune projects that I'll talk about. And then Easton Ford is a PhD candidate. He does less on the service end, more of a hypothesis-driven science project. But part of his project is developing a novel assay that I hope then once is validated can then become an option for service for anyone who may be interested. So I know that Dave already went over sort of all of the different applications for that PacBio can offer, but I do want to just sort of briefly mention again that the platform is incredibly flexible and it can take input that either derives from DNA or RNA that's been converted into cDNA. It can take long pieces like those 50 KB CLR libraries and down to small amplicons of 250 base pairs if you need. All of those things go on that Sequel IIe, which generates between 4 and 5 million molecules of sequence per run at very high quality, as Dave's already mentioned. And something that I've been really impressed with and is very useful as a core manager is the fact that there's constant addition and improvements to the back end accompanying analysis suite called SmartLink. So we're currently using SmartLink 10 that corresponds with the IIe instrument, and tools built into that user interface that are very intuitive and easy to use include assembly tools, mapping tools, variant calling tools, minor variant or haplotyping tools, as well as isoform prediction from the IsoSeq algorithm. Squanti for annotation there is in parentheses because it is a third-party tool. It's not packaged in the SmartLink software. Something I do want to call out particularly is the assembly algorithm IPA that was just released along with SmartLink 10 as part of the IIe upgrade. This has been just mind-blowing to me as a molecular biologist. I can generate those libraries and get you your HiFi data, but assembly was always something that was beyond my reach. It was something that was command line driven, took excessive amount of compute powers and maybe a month to do using some of the older algorithms. Building IPA into SmartLink means that I can do a preliminary draft assembly with these HiFi reads without much informatics expertise. That is an incredibly powerful starting point for any of our downstream collaborators who are doing annotation. The tool is ploidy agnostic, so it does provide phased, diploid, assembled genomes, in our case, for an equine species. And it worked in about 8 hours, which sort of blows my mind. I started it when I left and it was done when I got back the next morning. So good on PacBio for getting those tools up and running. I think it really helps flesh out the full suite of capabilities that the sequencing really brings to the fore. So what does our business model look like? So we launched our Sequel on November 16th. So we've only been live for about 4 months. We've sequenced 24 SMRT cells thus far. Most of them HiFi whole genome sequencing and a lot of internal assay development. That's about 6 weeks of solid sequencing. So if you count Christmas and a week for COVID shutdown, that's not so bad as far as uptime. If we look forward to the current queue, we have 36 SMRT cells in the queue. a combination of, again, of HiFi whole genome sequencing across a number of different species. A lot of IsoSeq is starting to come in, and then a couple of these assays that we develop in-house or are newly released from PacBio or partners. So in particular, tomorrow we'll launch our first 16S run using the strain ID kit that Dave mentioned. We're doing a 96 sample pilot. We're quite excited about seeing the quality of that data after recent data presentations and various webinars that PacBio has hosted. We also have our first set of HiFi viral samples coming online. We are planning to do our HiFi or COVID sequencing with 384 samples per pool. So we've got 2 pools already lined up and ready to go. And we were lucky enough to partner with PacBio for the recent HiFi for All Smart Grant. So the call closed a couple weeks ago. We've been evaluating applications, and we're very excited to announce the winner soon and participate with that sequencing project for the Americas region. So I think, you know, we are excited and happy to work with any of you, either collaboratively or as a service, or even just sort of advising if you're thinking about bringing PacBio to your campus, with any of the applications that are provided as sort of end-to-end solutions by PacBio. But I think one of the things that we offer that's a little bit different than a lot of cores is a lot of novel assay development. So one of the things that I do when I meet with investigators is to try to, you know, play that mental game of like, what can I bring to the table? How can I make smart sequencing work for your project? And so what I want to talk about briefly before I wrap up is a few examples of how we've done that on different projects, just to get the sort of mind moving on how this might be a good use for you or your customers. So the first example I want to give is this custom targeted IsoSeq example using the PIK3CA gene. So in this method, you generate total cDNA using the IsoSeq prep kit, and then you come in with gene-specific primers for second strand synthesis to make a targeted amplification of your gene of interest, but in RNA space. So that's critically important. So the gene that we're looking at here, PIK3CA, encodes the PI3 kinase, and 12 to 15% of cancer cases are known to contain multiple mutations in PIK3CA. These are SNPs that I'm talking about, and these SNPs have been identified by Sanger sequencing panels, essentially. We designed an assay to target this gene in order to try to understand whether multiple mutations in a single individual are happening in cis on the same strand or in trans, where one SNP is on allele 1 and the other is on the opposite allele. I'll tell you why that's important in a minute. Most importantly is that we did have to target it in the cDNA space. PIK3CA has quite a few large introns, and in the DNA space, it's about a 35 to 40 kb gene. You can't target that with PCR, it just really won't work very effectively. However, after splicing, it's about a 3 kb transcript. And that is really, when we started this project in 2017, that was an amazing sweet spot for this HiFi read generation, CCS reads that gave you very highly accurate sequence in order to be able to confidently call SNPs and look at this cis versus trans association. So we did that using the minor variant analysis tool. This is actually taken directly from that tool. These are 6 individuals. The specific mutations that were known to be in these individuals and were looked for in the PacBio data are on this left-hand side. And the presence of them in trans is represented by 2 red boxes. So if it's a mutant allele and it's a mutant base on 2 alleles, it's represented in red. So the SNPs that are in cis are in red, whereas the ones that wild type is in gray. And then the SNPs that are in trans are in either yellow or green, where each horizontal line represents a unique allele. And so what we were able to see with this assay was that a number of these individuals carried very high percentages of these mutations in cis in their DNA. And in combination with the functional data that Neil Vossen generated at Memorial Sloan Kettering, he observed that when these mutations were in cis, these individuals showed increased PI3 kinase activity, which unchecked leads to further proliferation and tumor growth. But as an increasingly active enzyme, it also made it a great target for PI3 kinase inhibitors. And so these individuals that also showed greater activity were also more susceptible to treatment. So the paper proposed that we use some type of sequencing assay like this to identify which individuals have those enzymes in cis and therefore may be more susceptible to really effective therapy using these PI3 kinase inhibitors. So it's just a really interesting and very clinically relevant way to use long read sequencing. The next assay I want to point out is this IGH genotyping you may have seen in my pie charts a few slides ago. This is something I've been working on with Corey Watson for, almost 6 years now. So Corey studies immunogenomics and understanding how each of us have a qualitatively different antibody response. And rather than sort of give people PTSD flashbacks to immunology class, I'll try to quickly go over this as painlessly as possible. Antibodies are encoded by a combinatorial recombination between a V gene, a D gene, and a J gene. What we don't know really is what the full extent of V, D, and J alleles and genes are in the human genome. So we currently have 2 references. This is a schematic of reference number 1 with each of these green boxes representing a V gene, these blue boxes representing a D gene, and these tiny yellow boxes representing the J genes. Corey, as a graduate student, generated the second reference and was able to add an incredible amount of genetic context to this locus. And just the difference between these 2 references already demonstrates a high level of variation in an individual to individual manner. Corey showed that greater than 50% of this locus falls within a large structural duplication, and that many of these genes are actually present in multiple copies in the same individual. There are extensive copy number variants. And that's where long reads really come into play. If you try to sequence this locus with short reads, often the reads that come off the instrument mismap or will not map to the correct variant because you have 3 alleles in a row, and those short reads don't quite know where they're supposed to go, and you don't get great resolution of this type of variation in the locus. So the first thing we did was use this data to justify the need for additional references. So both of these sequences were derived from either a haploid hybridiform mole or a collection of BACs and phosmids that were known to map to this locus, so no true diploid individual. So the first thing we did was do that. We used phosmid libraries generated in the Evan Eichler lab to sequence this locus across a number of additional individuals from different populations. And so this is schematic of just the 2 references I showed you, HG19 being the older reference, HG38 being the new reference. You can already see some macroscopic large structural variants that differ just between these 2 references. To note, each one of these boxes represents a V gene, and the color of that box represents the number of alleles in that V gene present in that reference. So in one case, you may have 1 copy of this V gene, but in this reference, you have 2. So these are the 6 of the 8 additional references that we've been working on. Again, 2 references from each of 3 different populations. You see, again, very dramatic structural variations that pop out quite immediately to just sort of a macroscopic overview. Additionally, you see the presence of these yellow boxes. So these were gene alleles that had never before been seen in the databases. And so we were able to identify many novel alleles here. I think what is striking to me as someone who's been studying viruses and vaccinology for most of my career is that there's a lot of vaccine strategies currently that are aimed at targeting the elicitation of a specific antibody response by generating an antigen that maybe say binds to V169. But if you look at V169 here, every reference has a different copy number or a different allele at this locus. And if you were targeting, say, V334, 4 of these references don't even have this allele. So trying to understand your vaccine efficacy without understanding the locus and the individual you're trying to stimulate seems like a challenging exercise. And so we argue that it's really important to know the context of your IGH to try to qualify and understand vaccine responses and immune mechanisms. So initially, this data generated a number of new alleles to put in the database, as well as an incredible amount of SNP variation that we went forward to parse, and I'll show you where that's important in just a second. We wanted to make sure that this assay would be available to be used with any clinical sample, not just from phosmid libraries in the back of people's freezers. And so we use this data to generate an oligo capture panel in order to produce a genotyping assay. So now we've done this genotyping assay on blood samples, on tissue samples. So this is something that is capable to be applied to most samples that you have. The strength of this is that we generated an oligo capture panel based on the variety of IGH context that we had from those prior references, and that we shear our DNA to 6 to 10 kb to generate libraries that would span a lot of those copy number variations in order to cleanly resolve structural variation. This allows us to get the IGH locus resolved in a diploid fashion. Oscar Rodriguez and Corey's lab, we printed this or published this paper last year, and he developed a novel software pipeline called iGenotyper, which produces a quite nice report, a genotyping report. And you can see both haplotypes resolved completely. Now, these are samples from the 1,000 Genomes projects that were derived from LCL cell lines, so there's quite a few genes that may have been deleted due to cell culture artifacts, but we do see the presence with these black triangles of a number of new alleles, even within standard reference genomes that have been characterized over and over and over. So as part of Oscar's paper and as part of our validation, we wanted to benchmark against standard short read methods that are used for resolving this area, this locus. And so using ground truth of the hydatidiformal CHM1 sequence, that was Corey's original project as a student, we compared the SNPs as true positive, false negative, or false positive, and the imputation and short read platforms definitely produce a lot more sequencing noise, as well as sort of a differential SNP count, where iGenotyper definitely pulled out far more SNP variation than some of the short read platforms. And the vast majority of those SNPs were in regions that were inaccessible to short read due to their sequence context. Now, what was really striking and what we're digging into now, it's quite exciting for me, is the impact of some of these SNPs on antibody expression, right? We're at the DNA level, but really what we care about is whether those antibody genes can be expressed as RNA and then of course as protein. Obviously, if you don't carry the allele, it's very hard to express that as an antibody. So here in this case of VGene1, if you have an allele that has a deletion or a haplotype that has a deletion and you're heterozygote or homozygote for the deletion, you don't express that in your antibody repertoire. If you're heterozygous for an insertion and deletion, you express lower levels than if you're homozygous for the insertion. However, what's been really striking is looking at these SNPs that are not in the coding regions. So these SNPs are not in the alleles themselves, but are upstream or downstream of the alleles in regions that may dictate the ability of recombination machinery to cleave and incorporate those alleles into the transcript that then becomes the antibody. And what we've seen indeed is that there are particular SNPs outside of coding regions that impact the ability to express this allele in the antibody repertoire. So in this case, this A SNP upstream of VGene2 is directly impacting, appears to be impacting the ability to express VGene2 as a transcribed allele in the antibody repertoire. And then if you're a heterozygote for A and C, you express lower levels of that gene. And then if you don't have that A, you express high levels. So we're really digging into the impact of these SNPs on the antibody repertoire and how that affects, again, immune responses and responses to vaccination. So with that, I'll wrap up. I'm happy to take any questions. I do want to make a point to, again, thank my lab, thank the Watson lab, thank my benefactors here at U of L, Ron, Harrison, and Kevin, as well as my collaborators at Sinai. That was just such a stunningly amazing experience. I'm so lucky I had it. My new horse collaborators at UK, as well as make sure that you all have plenty of time to see my contact information as well as our website. Don't hesitate to reach out. I would be delighted to speak with all of you. So with that, I will stop sharing and be happy to take any questions.
Thanks again, Melissa. I mean, every time it's just an amazing body of work, and just really appreciate you taking the time to share it with everyone. So I know we have a moderator who's going to help us with some of the questions that came through. So yeah, let's see where we're at.
Awesome. Thanks, Melissa. Wonderful talk as usual. I did want to remind everyone, if you have any questions, please go ahead and put those in the chat function. We did have a couple come through, and one of them is kind of around, you mentioned that you guys are offering quite a few applications on the PacBio system, and with being a new lab that was just established, can you address how hard it was to get new scientists in your lab up to speed on the wet lab side, and to be able to offer these applications with good repeated success?
Sure. What I love to be able to say, and it's not at all euphemistic, is that the PacBio library preps, the molecular preps themselves, are by far, I find, the easiest preps amongst all of the NGS systems, and so if you have someone who's done molecular biology before, they've done restriction digest, they've done ligations, they could do a PacBio library prep. It's pretty straightforward. I think really the hardest learning curve is some of the ancillary equipment, using how to use a blue pip and well, and these types of things. But even then, the support staff at Sage or Agilent, in case of the QC equipment, is incredible. I have admittedly been incredibly lucky with the people I've gotten. They're very talented, and I certainly don't want to take any of that away from them. But the support that PacBio provides for training, for questions, and to help me get my people up to speed has been incredible, and it's been far less of a challenge than I anticipated.
Awesome. Yeah, good hands are always really, really nice to find.
Yes.
Okay, I think we have time for one more question. So you did kind of address this a little bit. You talked about how upgrading to the Sequel IIe was really helpful in terms of data storage. Can you maybe go over a bit about analysis and interpretation and how that is a bit more straightforward with HiFi data, and just
Sure.
how that helps you deliver data to
Right.
customers a bit easier?
Right. I mean, data delivery is just a breeze now. There are projects I can deliver via personal Google Drive that I couldn't have done, that I would have needed an S1 bucket for before. Just the data size alone, especially for transfer and delivery, especially when you're not local or don't have a high-speed line. I mean, there are things like that just, it changes everything. For analysis, it's also fantastic. I mean, I think, a lot of the pipelines, well, almost all of the pipelines now, right, have that HiFi generation or CCS algorithm up front. So IsoSeq, assembly, these things. And so having that done on the system in the background, right? So HiFi generation starts when your first cell is done while your second cell is running. So your data is running off the instrument in a way that it is ready to come out onto SmartLink and be analyzed through any downstream application, which means that an IsoSeq job that used to take 48 hours on my server now takes 2, because the bulk of that was CCS generation, and now it's done on the box. And so it streamlines, it speeds up, and it compacts everything. So overall, all of those factors make everything much, much more efficient for us. And honestly, I want as few things waiting on me as possible. So to be able to build in automatic demux, automatic HiFi generation, automatic export onto the box means that the only thing waiting for me to push a button is any downstream tertiary analysis beyond that. And frankly, a lot of people just want the HiFi reads. They want to play with them themselves. So that means I don't have to do anything. I just have to export that data. So that's a pretty incredible perk, because I don't want to be the person responsible for getting you your data late.
Yeah, no, that was a great explanation. Awesome. Well, it looks like we're just about up on time. So thank you so much again for presenting. And if anyone has any additional questions, please come visit our booth tomorrow. We'll be around for the rest of ABRF, and we'd be happy to chat.
Thank you.
Thanks, everyone. Enjoy the rest of the day.
Yes.